So let me welcome you to the October 21 version of the seminar series of the IMAG-MSM Working Group on Multiscale Modeling and Viral Pandemics. As always, this meeting is live streamed on YouTube and is being recorded. So don't, for the speakers, don't put anything up on your slides you don't want to have broadcast to the world. If you move on to the next slide, Bruce, just to remind everybody who has not been here in a while, the co-leads of the working group is myself and James Glazier, together with Jim Sluka and Bruce Shapiro. You've heard from both quite a bit. Any questions, any suggestions for speakers, please contact one of us. We have a Slack channel, we have a Twitter feed, we have a wiki page, we're on LinkedIn, we're on YouTube. So, you know, knock yourself out connecting to us and please use all of those. Any, as always, we have a minute or two for any short announcements that anybody might have about meetings, funding, job opportunities, publications, conferences. Anybody would like to make an announcement? Okay. Okay, nobody has any money, huh? All right, so just to give you a highlight of upcoming talks, next week we have Ross Taylor from Johns Hopkins, David Forgach, University of Georgia, and November, Ulrich Schwartz from Heidelberg talking on malaria, Stanka Chupi from Virginia Tech on COVID-19 testing protocols. And for the rest of the year, we're actually quite booked. So we have interesting and interesting speakers for all of those weeks for the rest of the year. Yeah, so if you could please mute your microphone. What we typically do is we keep all questions until the end of both presentations, but that works only if both speakers are able to stay on potentially after 4 o'clock if there are questions still for both speakers. Otherwise, we'll take questions both after the first and the second talk. So Jason, I think you were the first one. Do you have time to stay on a little bit after your talk?
Yes, I can stay on.
You can stay on, terrific. And same for, sorry, I lost now who the second.
Yeah, I would be able to.
Terrific, thank you very much. So we'll just move from the first talk right to the second one. And I think this is the end of announcements, is that right? Yes, so let me introduce Jason Shoemaker, University of Pittsburgh, who will tell us about some of their work on COVID-19. So go ahead, Jason.
Okay, thank you. Let me go, oops, I can't share my screen yet. Oh, there it goes. Green. Okay. Are you able to see my slides?
Yes, we can see.
Right. Thank you. Okay, so thank you for having me today. I scheduled only about a 20-minute talk because I think that's what the online guide said, to aim for about 20 minutes. So if that wasn't long enough, if you want to do questions earlier, I'll leave that up to however we would like to proctor this talk. But again, thank you for having me today. My group's been doing a bit of computational modeling now for a little while with influenza, COVID a little bit now, and herpes. And I'll talk to you today about some of our projects. And just quickly to give a quick background then, I do need to update this slide, so I am missing at least one student. But we typically kind of work in two big areas on the research side of things, and then some ancillary areas. So multi-scale modeling, essentially what our MSM group here does for specifically currently viral infections. And we've also done a bit of work on big data algorithms using large scale protein interaction databases and trying to infer drug targets and predicting mechanisms as well. So I'll briefly give an example of each one of those groups today in our research talk today. But we've also done some other things. Of course, we're in engineering, we're always trying to do education and outreach. So we have a small component in my lab that has worked on trying to do some VR education related to virology. So we have produced a game that's available. It's still on Apple devices, doesn't work anymore for Android devices, but allows people to try to, especially for kids, to try to explore the body as if they were a virus and try to infect cells and try to learn about how the cells detect a virus and then respond to an infection. And we also have done a little bit of work on helping with design some medical devices. So with that, then specifically in the modeling products, we've had quite a few products come out in the last few months and the last few years also. And I'm going to focus on only one or two of the products today, but I wanted to highlight these because I know we've had a lot of overlap in our community here with a lot of presentations that happened at, like, for example, the Fred Hutchinson virus workshop that was about 2 weeks ago. So specifically then, we've done a lot of work on strain-specific immune responses. So looking at H5N1 viruses versus H1N1 viruses and trying to understand why do they seem to induce slightly different immune-looking dynamics, whether or not there are actually kinetic differences leading to those responses or not. And I'm not going to talk about that today because I've been told from Emily, my former student who's now a postdoc in Harvard, that she has been asked to give a talk in the next couple of months actually with the IMAD group. So she'll be giving a talk on that section then in about maybe a month and a half or 2 months from now. The other area we worked in quite a bit then has been mathematical modeling of RNA viruses in general. So this is much more trying to look at the RNA signaling pathways. And that's work that we've actually done in collaboration now with James Glacier, TJ Sego I saw was on here, and also Josh and Jordan. And this work has now been accepted, so hopefully it'll be on, the preprints are already in the bioarchive, but hopefully the actual version will be out soon. We've got the official acceptance now. Now we just have to go through and make sure the templates are all correct, and that'll be up and loaded. So this one was presented and heavily discussed at the Fred Hutchinson virus workshop. So a lot of individuals who are particularly on this call, I recognize from that workshop. We already had a lot of communication and dialogue about that research as well. So I decided also not to talk about this one today, because again, it's only been 2 weeks since we've seen that. So today, what I'll focus on in terms of the modeling side is going to be our DNA signaling components that we've been working at. So looking at the C-gas pathway specifically, which is a pathway known for detecting viral DNA, and also just DNA and cytosol in general, which is always kind of a bad thing to happen, and how we use differential equations and agent-based models to understand how those signalings behave. So the two specific projects we're going to look at then today are one on I see here on the left-hand side is that we'll be using agent-based modeling and try to look at some different behaviors that we can see that can occur in signaling given certain characteristics of signaling. So specifically want to see how does stochastic behavior either improve or inhibit robust immune responses. We also looked at cellular heterogeneity in this particular study to see is there an advantage to being heterogeneous or is there an advantage to being more homogeneous? What is the design implications then of these kind of behaviors that we know to be a factor in interferon signaling? The other thing I'll talk about briefly then is some of the virus host subnetwork construction that we've done. And in this particular project, we've taken gene expression data and protein-protein interaction data, found different ways to overlay them on top of each other to try to identify host proteins that are important for virus replication. And of course, the logic there is if that protein is important for virus replication, it's also a reasonable target for drug development because then you can inhibit viral growth. So we'll start with the first project here on the left then. And on the left, you can see here, sorry, my slides are a little messed up on my screen here, but immune signaling interferon production, this has been known to be stochastic now for quite a while. So this is work from Tom Maniotis' lab. And you can see that as they take cells, different cell lines, 3 different cell lines here, and challenge them with a particular, either a virus or immunostimulatory DNA or immunostimulatory RNA, there's lots of different ways to activate interferon signaling, that only a fraction of the cells typically turn on. And you can see they measured this dynamically over time, and at any point in time, only about 15% of the cells in the cell culture seem to be actively producing interferon despite a pretty robust simulated infection. So the question really becomes then is, what is the advantage or potential disadvantage of this kind of stochastic behavior that we can observe in the stochastic signaling of interferon production? So to address this particular matter, my student, Rob Gregg, developed an agent-based model of specifically the herpes infection, so DNA viral infection, to determine what kind of advantages might exist if you have a high stochastic signaling environment or highly heterogeneous cell populations. The particular model was developed in Julia, uses differential equations to describe each one of the cells' intracellular signaling, and that intracellular signaling itself was trained to data from immunostimulatory DNA studies, specifically in fibroblast cells. And then, of course, we use partial differential equations to help model the diffusion of virus and diffusion of interferon across essentially a simulated cell culture. And then beyond that, we added on some other additional levels of complexity. So we also started, as we can see here with bullet point B, we considered heterogeneous initial conditions to simulate cells that are in different states. They have different levels of proteins during the initial stage of infection. So cellular heterogeneity, we can kind of tune that in this particular model. And stochasticity, same thing. We were able to tune how many cells turn off and on by basically controlling one of the parameters that controls the production rate of interferon. So with a very direct method, we're trying to say, if we can tune the stochasticity or tune the number of cells turning on to produce interferon, was there an advantage or disadvantage to having a certain number of cells doing that? And then for completeness, we also had to, of course, include some other factors such as transition from a healthy cell to an infected cell. How do we model that? Was mostly based on a probability equation based on the how infected your neighbors are. And we also developed an equation about how you transition from infected to dead, based essentially on how long you've been infected. There's different ways we could have considered that, and I'll be happy to talk about other ways to model that later on. Hopefully you all saw those slides. I may look at the wrong slide. So the first thing we wanted to show is that we were able to capture the signaling dynamics accurately in this particular system. So you can see here that we collected data from 1, 2, 3, 4, 5, 6, 7 different studies, and used these to help confer whether or not our model is doing at least reasonable dynamic behavior. Of course, the data is incomplete, it's not precise, it's not perfect. Some of the data is protein and some of it is absolute concentration. So some of this is nanomolar concentration data. Some of this, specifically like the mRNAs, are qPCR data, so it's relative fold change data. And we actually just scaled these to what would be a reasonable peak based on other pieces of evidence. But in general, we pretty much get a pretty solid prediction between our model and what has been observed in cell culture. And this is not tuned at all. This is taking a cell-based model that's using ISD, so immunostimulatory DNA, to predict the behavior of interferon, and just putting it into this multicellular environment. And we pretty much are able to replicate what we observed then, for the most part, in vivo. Something we're able to do that we couldn't do experimentally, of course, was consider primary cells versus secondary cells. And in this particular work, we define primary to be cells that are directly infected with virus. How much interferon are they producing? How much interferon mRNA are they producing? So we can see these lines here in red. Whereas we can also consider secondary cells, cells that are not directly infected by the virus, but are actually being impacted by interferon's paracrine signaling. And that's what these blue shaded regions are. These represent the behavior of neighboring cells that are not actually infected, but are still responding to the infection itself. So overall, we were pretty satisfied with the model, at least being able to experimentally to replicate experimental data. And that's when we decided to dive in and start doing some analysis of the model. So the first thing we wanted to explore was cellular heterogeneity. And the question we wanted to ask ourselves here is there an advantage to having highly heterogeneous cell populations or a disadvantage? And the way we tried to model this then is we took the initial conditions of all of our cells in our culture and allowed those initial conditions to vary based on a particular variance, seen here on the x-axis of these two plots, from 0, so no heterogeneity, completely homogeneous cells, all the way to a standard deviation of 1 from their initial value. So what this allowed us to do essentially then was to artificially create environments of very heterogeneous cell cultures versus homogeneous cell cultures. And what you can observe here is, as we allowed the heterogeneity of the cell culture to increase, you see that you're actually able to get a more robust production of interferon for cells that have higher heterogeneity. So one of our conclusions was, and this was true whether we were letting the cells be deterministic, where almost all the cells can produce interferon, or whether letting them be stochastic and only part of the cells can produce interferon. So what this kind of told us is at least heterogeneity might be a way to actually kind of tune or enhance your interferon production capability in a cell culture. So that was one thing we wanted to look at, but of course we were more interested in looking at stochasticity because we wanted to understand why was it designed to have some cells producing interferon and others not. And that's what we saw here. It was, we wanted to ask, is there some kind of benefit? And defining a benefit is always a challenge. And we decided to define a benefit as being what are conditions that lead to minimizing the number of dead cells. So we see here on our x-axis is the percent of dead cells, and what we're seeing on our y-axis. What we're seeing on our x-axis here is us artificially tuning the number of cells that are stochastically turning off or on during the course of the infection. And as we go from 0, so all the cells just do not produce any interferon, so of course 100% of your cells die, all the way to 100%, so 100% of the cells are producing interferon, so this is essentially not stochastic response anymore. We can see that there's definitely initially from 0 down to about 20 to 30% of the cells responding, producing interferon, there is a suppression and a drop in the number of dead cells that resulted at the end of the simulation. However, if you keep tuning that number even higher, if you allow more and more cells to respond and more cells to produce interferon, then we didn't really observe any benefit anymore in terms of the number of dead cells in our total simulation here. So our takeaway from this was, just based on this model that we constructed, again, that we didn't do any real training on other than fitting it to some fibroblass data, that very interestingly, it's telling us that number they observed experimentally in the Manos experiments of about 20 to 30% of the cells stochastically turning on during the course of an infection also correlates within our model of this kind of break-in point we could see where after about 20, 30% stochastic signaling, there's really no gain into making more cells turn on in our environment. So this was just kind of a fun thing for us to look at. And we thought this was pretty exciting. So this was our initial foray into agent-based modeling. And now we've done a lot more work with this now with James Glacier, some work with Paul as well. So hopefully then in the next couple of weeks, when one of my students, Emily, goes to present later on in the IMAX series, you'll be able to see some more of the newer studies that we're doing that are coming out pretty soon. So the quick takeaways here was that agent-based models allow us to consider spatial effects, stochasticity, and heterogeneity. And again, the thing that we were most interested in is this bullet point right here that I'm pointing at with my arrow. That stochasticity limited the number of cells that really needed to produce interferon in order to minimize cell death. And we think that's a very interesting observation. So that's the end of the first half of my presentation. So the second half of the presentation is going in a very different direction. And we're looking at trying to identify antiviral drug targets. And this is something that's just desperately needed. Obviously, with the current state of the pandemic, I don't really need to sell this point very hard. But before the pandemic, if you were looking at flu specifically, which we're primarily interested in, there's not a whole lot of drugs that are really effective antiviral drugs for flu. Osimetafir, which better known as Tamiflu, is also robustly just not very good. The viruses, that when this came out about 2008, 2009, viruses adapted pretty quickly and were able to escape these osimetafir's effect. You have zanamivir and peminavir, but those also have some considerable limitations. The one for peminavir here that is particularly concerning is it's not recommended for children or people with breathing problems, AKA your high-risk group, the people you really need to be treating. So we do need to find a way to try to identify more exciting ways to find antiviral drug targets. And the way we tried to do that was by looking at influenza proteins and looking at where within the host proteome do influenza proteins interact with human proteins. So we collected a whole bunch of data where we took the human proteome, shown here in black, and then identified all of the human proteins highlighted here in blue that are known to interact with different proteins of the influenza virus. So you see these proteins here, these big gray circles, these are all influenza proteins. So you have your NA, PB, M1, all these different proteins that make up your standard influenza virus. So you end up with tens of thousands of interactions, a lot of proteins involved. And the first thing you always want to do when you have this kind of a big hairball to analyze is, let's look at things like the number of degrees, the number of interactions a particular protein might have, the betweenness of these human proteins here in blue, to try to see if they're really important to signaling across all this entire network, and try to see, is there some interesting differences occurring or not between these two families of proteins, the ones that directly interact with the virus versus those that do not interact with the virus. So when you do that, you can see we have our virus binding proteins here in red, the statistics related to them, and then all other proteins that are not viral interacting in the white data shown here. And we have our degree on the left-hand side and our betweenness on the right-hand side in the B panel here. And yes, there are statistical differences between the degree and the betweenness of virus interacting proteins than there are for standard human proteins. But you can also look at this and say, that, yeah, there's a statistical difference between the mean and the median of these two populations, but there's also a ton of overlap. So you can pretty quickly tell with degree and betweenness alone, you're not going to be able to very confidently go ahead and say, okay, I can choose protein X based on its degree to be more likely related to virus interactions and maybe have some kind of role in actually affecting and impacting viral replication. So we wanted to try to take the analysis a step further. And again, the takeaway here is that topology distinctions really don't seem to be able to predict host factors very well. So several studies have suggested that interaction data can predict protein function. And we wanted to try to make this something a little more clear cut than just looking at in a broad sense betweenness and degree. We wanted to make it much more focused and localized. So what we did is we took host virus interactions and created a system to mine the human proteome in order to identify all the closest linkages between virus interacting proteins. So again, emphasized here in blue in this figure, and proteins that we call internal essential host proteins. This was a group of about 25 proteins that has been shown in multiple studies to be very important for virus replication, not necessarily good for drugability because of side effects and other concerns, but definitely known to be very key for viral replication in general, and not specifically just one strain of influenza virus, but multiple strains of influenza virus as well. So we decided, okay, let's take our virus interacting host proteins. We've identified a set of internal proteins that we're very interested in, and let's take the closest avenues, the closest connecting routes we can to connect those through our human proteome, try to find the shortest paths. Take all potential shortest paths and extract what we call our extracted subnetwork over here on the right-hand side. And the idea was, okay, if we can take this and really isolate this localized part of the network that might be really, really related to influenza viral replication, maybe we can identify zones that are enriched for potential antiviral drug targets. So we decided to analyze them. We also looked at the betweenness and the degree of that subset of proteins that we identified within our subnetwork. And one thing we observed pretty quickly was that while degree of the proteins are identified in our virus subnetwork versus their position in the standard all human PPI, degree really didn't change very much, but betweenness changed pretty largely, I would say, between the human PPI position and this subnetwork position that we identified. So we decided in that particular case, let's go ahead and use betweenness as an additional category to help prioritize candidates within our virus host interaction subnetwork. So to wrap this up then, we have this hairball that we've then extracted and got to a nice small localized network. We're taking the proteins in there and taking the ones with the highest betweenness. And we're going to say, okay, we're going to hypothesize that those might be highly enriched for antiviral drug targets. And the way we tried to validate that was by doing an siRNA screen. And this was done with my collaborator's lab, Yoshi, in Wisconsin. So as a reference, hopefully you can see this better than I can because this thing's getting in my way. As a reference, we have a whole genome screen, which tells us what is the likelihood of finding antiviral drug targets by random guessing. And it's a pretty small number, somewhere around 1%. So we have that as a reference for just random guessing. From these viral interacting proteins, there was a study that took all those viral interacting proteins that show in the very beginning, did an siRNA screen, and they found about 20 to 25% of those proteins tended to be really promising looking targets for antiviral drug consideration. In our system, when we looked at just the subnetwork, so randomly selecting proteins from the subnetwork we identified, we got up to about 35% as a hit rate for potential drug targets. And then we had added on that additional category of being within the subnetwork and being high betweenness. Then we got close to about a 40% hit rate. And just for completeness, we also sampled within the subnetwork and low betweenness and showed that, yes, that was actually somewhat pushing down our performance in terms of identifying potential drug targets. So in the summary, the takeaway, because apparently I am taking more time than I thought I would, the takeaway here is that by doing this subnetwork approach, we tried to isolate a local part of the network, and then prioritizing based on between this, we were very able to identify a very enriched area of potential antiviral drug targets. And with that, I will wrap up that particular part of the talk then by saying integrating virus-host interactions, screening data on network topology can definitely improve our antiviral drug discovery. This particular approach, also something I didn't go into, I skipped over that slide, was that the subnetwork approach actually allowed us to start seeing certain enrichment of particular factors we want to see within our network as well. Because when you looked at just all of those human proteins that are known to interact with the virus, you don't see any really interesting Go categories or anything like that. But when you look at the subnetwork we identified, then you start seeing immune signaling appear as being very enriched within that particular subcategory. And the last bullet point here, I actually didn't talk about today, so we'll leave that there. And anything related to this particular way, here's the 3 publications for those works right there. And with that, thank you for the time, and I will stick around later on for any questions.
Terrific. Thanks very much, Jason. Great talk. So we'll hold questions until after the second talk. So let's move on right away to our second speaker, Tiam Hadari from the University of British Columbia. It's all yours.
Can you see my screen?
Yes.
Okay, awesome. Hi, everyone. This is a privilege to be here. My name is Tiam Hadari, and I'm a PhD candidate in ZASA Lab at the University of British Columbia, School of Biomedical Engineering. We are an interdisciplinary lab who work on stem cell bioengineering. So we are combining expertise from stem cells to bioinformatics. Today, I'm going to talk about our platform called IQCELL. And our platform is a platform that basically generates a gene regulatory network for developmental systems from single-cell RNA-seq data. Basically, the gene regulation and development are really connected, and the root of studying gene regulation actually is in development. Starts from early 1900s, from works of Theodore Bowery, when he basically observed that the blueprint of development is located inside the chromosome. A little bit coming forward in 1930s, Hunt Morgan showed that, or hypothesized that the stem cell differentiation is due to differential gene expression, which was a very important hypothesis in the field. And then again, a little bit flash forward to 1960s, Eric Davidson and colleagues for the first time conceptually connected the gene regulation to stem cell differentiation. And basically, the promise of this field of connecting the gene regulation to differentiation is to being able to predict the effect of gene perturbations on the outcome of differentiation of stem cells. When we are thinking about this prediction, we need to consider what type of interactions we are interested in. Basically, I would encourage you to read this nice book from Judea Pearl. Basically, there are different levels of hierarchies of relating objects together. For example, you can start to think about correlating objects together. For example, 2 genes, when you want to think about how these genes are related, you can say gene A and gene B have some sort of correlation. And this correlation basically gives us some sort of a statistical relationship between these 2 genes. But if you want more detail, of course, these genes can have actual some sort of causal relationship. Gene A or gene B can be the effector of the other gene. So another level of this hierarchy of interaction or causality is of knowing the direction of the interactions. What's the direction of the interactions? When you know the direction of interactions in something like a gene regulatory network, as you know, you can predict some effects in a local level. So for example, you can say that if I perturb gene A, this should happen to gene B up to some level. But if you are interested in knowing the effect of gene perturbation in a global level or at the system level, you prefer to have more information about these interactions. Basically, you need to have some details about these interactions that then can help you to first predict the effect of perturbations in system level and also help you to implement forward engineering thinking. So design circuits and then implement into your cells. And that's basically the goal that we have in this work. During these years that this field basically developed, many people try different ways to implement and obtain this type of networks. The main approach have been this closed loop of genetic perturbations, observation, and then model development. And then when you develop a model, based on the model that you have, you can now predict new perturbations, go to lab, and then basically run this closed loop for many rounds, and then you end up with a gene-regulated network for your developmental system. And the very, very prominent example is the work of Eric Davidson and colleagues, which actually the complete piece of it published at 2012 at this paper from Peter et al., when they showed that the gene-regulated network that they developed through the years, if you use a Boolean paradigm to just assign any gene to be on and off and having some Boolean interactions, it can predict all the events during the early differentiation of sea urchin. But this has not been the only way of thinking about developing gene-regulated networks. In this classic paradigm, for example, a prominent work of Sarah Jane Dunn showed that you can basically break this loop and make a pipeline of it. It means you can start with some random genetic perturbations. For example, in this case, in this paper at Science magazine, they run many perturbations like knockouts, knockdowns, and overexpressions. And then when they combine them, they fed it into some sort of automated reasoning machine from Microsoft Research. And then they obtained a minimal gene-regulated network for the system. So of course, at the end of this pipeline, you can go back and predict some stuff, but you don't need to run that loop for many rounds. But as you can see, these type of methods are expensive in terms of cost and time, because you need to go to lab and run these experiments multiple times. New sources of data, like basically that are rooted in single cell sequencing, actually have gave us an unprecedented amount of information that people try to exploit and use them to obtain gene-regulated networks. I encourage you to take a look at this nice review about the current methods that people use to obtain gene-regulated networks directly from single-cell RNA-seq data. At a nutshell, single-cell RNA-seq is basically you dissociate the tissue that you have, and then you send it to your next-gen sequencing machine, and then it gives you, for each cell, it gives you a metric of expression levels of all cells in terms of the messenger RNAs. And then you can use this information to also generate, if your system is a differentiating system, you can generate a trajectory for your system along differentiation. So that's what's happening in this type of data. And we are particularly interested in a very certain kind of a network that we can obtain from this type of information. We want our network to be predictive at the level of gene-gene interactions. It tells us what's the relationship between genes, and also we want it to tell us about the effect of gene perturbation at system level. So we don't not only want the correlation and directionality, also we are interested in the information like about the interaction details in our system. And to do this, basically, we developed the platform called IQCELL, which is a platform that predicts the effect of genetic perturbations on developmental trajectories by basically generating a gene network. The input of IQCELL is you give it basically the expression data from your single-cell RNA-seq data, and also inferred pseudotime. And then IQCELL has a part that it can basically select some genes that looks to be more informative, but you can also add your genes manually that you think are informative for your system. Then it starts basically make a very dense correlation network based on genes, based on mutual information between them in terms of expression. And then it starts to basically use a hierarchy that it infers from the genes to make a causal arrow between the genes. So at basically directionality to them. And then when your network is directional, but it's very dense, we use a platform called Z3 reasoning engine from Microsoft Research to infer Boolean network from this data that can represent how is the developmental trajectory during the system. And then IQCELL also has built-in simulation part that helps you to simulate your Boolean network and compare it with your actual experimental data, and also perform perturbation in your system and study how they relate with your system. So in the next part, I'm going to basically walk you through an example that we have used to basically demonstrate how it works. So as an example, basically one of the interests in our lab is T-cell development. And we are interested in development of T-cells from induced pluripotent stem cells. So that's why we basically tried mouse T-cell development as our model system. mouse T cell development mainly happens in thymus. So we have some progenitor cells that usually migrate from bone marrow into thymus. And then inside the thymus, there's a notch signaling, very strong notch signaling that starts to walk these progenitor cells from early T cell progenitors towards the differentiated step. So start from ETP, then they go to double negative 2A, double negative 2B, and then double negative 3, and then they basically go to double positive and later selection events that happen. Our part of interest and the part that we have data for, and we want to test IQCELL against, is inside this scope that the notch signaling is active. So we use the single siRNA-seq data that has been already published at 2019. And basically, we know that LMO2, for example, is a marker. And this is basically UMAP of the single siRNA-seq data. It's something similar to PCA, but a little bit more nonlinear. LMO2 is a marker of early T cell progenitor, and you see it on the left-hand side, and then CD3E is a marker of the N3 stage, so you can see that the right-hand side. And it basically, by testing all the genes that we know, you can infer that development comes from the left-hand side to the right-hand side. There are multiple platforms that you can basically put your data in, and they infer a developmental trajectory for your system. It means they basically group the cells to each other, and then they infer a pseudotime for your development. And we use a couple of them, for example, Monocle to infer a developmental trajectory for our system. So it starts from the left-hand side and goes to the right-hand side. It means that each cell now has a pseudotime point that measures where it is in this developmental scope. And now we basically take these two pieces of information, the expression matrices and the developmental trajectories, and feed it into IQCELL. So first thing that IQCELL does, because IQCELL basically is based on Boolean paradigm. So it basically assigns, and this is basically a hypothesis that we assign to each gene to be either on or off. First is a start for each gene and looks at the expression levels and use K-mean clustering to discretize the expression level of each gene, and then use this discretized expression and basically smooths the matrix. Then we start to making our initial gene regulatory network for the system. The first step, we calculate the mutual information, which is some sort of a generalized version of correlation between all genes. And then we make a very dense network of interacting genes. And this network is based on the amount of mutual information. Then we use and then we measure correlation between each gene. And if it is significantly positive or negative, we assign a positive or negative sign to this interaction network. And then we use basically elbow method to cut off the interactions that don't look to be significant. And that's how we ended up with a network that is a little bit less dense than a small word network, but it is of course still very dense. Then we use the hierarchy of genes that we infer from the data. How we basically infer this hierarchy is from these transition points that we infer from the data, where the gene starts to be from on or off to on. We call it the transition point. And then we sort the genes based on this transition point and then make a hierarchy. And every gene at the higher level of hierarchy can basically influence the genes in the lower level of hierarchy. And we basically end up with such a directed dense network to start with. Then we take this network. This network is really dense, and the only positive information that it has is basically how much the genes look to be related to each other. Because we use mutual information, this relationship can be really nonlinear. Then we use Z3 engine. Z3 engine is basically a reasoning engine, and what it does is basically you tell it two conditions, and then you ask what can be a logical transition between these two conditions. What we do is we take the pseudotime of our data, and then we ask Z3 to tell us the rules for each gene. What can be the update rules for each gene if you want to go from one cell to the next cell and this cell to the next cell? So we ask it to basically fit a nice gene network in a discretized way. So it's a Boolean network that can explain all the data that we have. And this data is already discretized by our K-mean clustering. When we do this, we basically end up with a gene regulatory network that is now both correlated and then it's directed, and also it has some interaction details. For example, when we look at the update function that we obtained for BCL11B, which is a very important gene in T cell development in mouse, we found this update rule with cells that GATA3, RUNX, and TCF7 also along with Notch1, upregulate BCL11B, and SP1 downregulates it. And this was very, very, very exciting for us, because this update rule, or this type of detailed interaction, had been developed during maybe 20 years of research in the field by Ellen Rothenberg and colleagues to know what is exactly the genes that are regulating BCL11B, but we can capture it now by one shot of basically exposing IQCELL to the data. In summary, there are 38 total interactions in our system, and we took a look, and we could find 29 of them are already reported in the literature. So we basically captured 29 out of 38 interactions that are there with IQCELL. But now that we have a network that it looks that the connections are correct, the next question that we should naturally ask is how its dynamics is working, because our network is Boolean. Now we can basically also explore the dynamics. So now here you are seeing the PCA plot of the data that I showed you, the single cell RNA-seq data. And the color is pseudo-time overlaid on top of it. So the blue means this earlier at pseudo-time, and the red is later at pseudo-time. Usually the cells start from commonly inferred progenitors or LET cell progenitors, and then develop towards the N3A in our data. And this is the natural flow of the data. And this is, by the way, discrete size. So we first discretize the data, and then we plot the PCA. Now we start our simulation for our GRN, and we start from LET cell progenitors. This is the step that we started. And the light gray basically is the experimental data. And then when we simulated our data, you can see the dark gray is our simulated point. So our simulation points are actually matching the flow of the data that we have in our system. So we start from here, and then this is going to be our attractor. If I want to be more precise, here you see the experimental data, the matrix along the pseudo-time. And this is the experiment, and this is the simulation data that we have. So we start kind of the dynamics, recapitulate what's happening in the experimental system. Not that we can have the dynamical system recapitulating what's happening in experiment. The next question that you're asking is how much we can predict about the genetic perturbations against the experiment. So we basically have a matrix, have a basically metric for measuring it. For example, if we want to see what's the similarity between our network and the experimental states that are established, we have a matching score. For example, our white top network, when we simulate it, starts from LET cell progenitors, and it ends up at the N3A stage. And when we look at the attractor, it matches 100% with the N3A stage. So that's basically what you can see here. Next, we basically run multiple genetic perturbations, and then we find the attractor, as you can see here, and then compare it with the state that we know are already there from experiments. And we found out that we can basically predict the effect of perturbations of gene regulation on developmental trajectory when we compare them with literature. For example, it's well known that when you knock out or knock down Notch1 in your system stops at LET cell progenitors. And when we do that in our simulations, you see that the system stops right here. So it doesn't proceed toward D2N or D2N, D2-3. Another example is BCL11B. It's very well known that when you knock down or knock out BCL11B in your system gets stuck exactly at the N2A state. And when we simulate this effect, we exactly see that the simulation stops here. One interesting example because these are all single gene basically perturbation, but we can also perform two gene perturbation at the same time. But not all of them, of course, is already have done in literature. So for example, one of them that have been done already in literature is when you knock down the Notch1, but at the same time overexpress TCF7. And by doing that, it has been shown that the system can go a little bit forward up to D and to A stage. And when we simulate that, we also see that one of the attractors actually is there. So you can see in the left side the experimental observation, the right-hand side, and the left-hand side the simulation. But the system that you're seeing here, basically the genes that we selected are at this point are manually curated. So we knew what are important genes because of the years of research in the field, and we just feed into IQCELL. The next question that we're asking is, basically, can we go ahead and predict some genes that are important, and then fit them into IQCELL, and how much our predictions are still robust against this set of genes that we are selecting. So we have a pipeline in IQCELL that basically you give it the expression matrix, and it uses a platform called Cynic or PyCynic. And basically what PyCynic does is goes and find basically during the databases what is the binding motif of your transcription factor to part of DNA. And if that transcription factor is active, and it gives you a list of genes that transcription factors or regulons that are important for your system, but it can give you up to 500 regulons. Next, IQCELL grab that data and then sort these genes based on the score. We call it dynamical time score. So it basically measures how much the gene is dynamic along the pseudo time. And then we grab the high genes with the highest score of GDS, or gene dynamical score, and we use those genes as informative genes in our system. When we've done this, we saw that basically we were able to capture 8 of the genes that we already know are very important and were in our curated list. And then also we have some new genes that can be basically tested experimentally later. But the next step, we basically we basically inferred another gene network based on this new set of genes. And then we compared the perturbation effect of the genes that we already knew from our curated list, and compared it with our new data. And we saw that from all of these genes, only SP1 is, the perturbation of SP1 is not exactly matching the data that we already have. And this is important because SP1 is known to be really masked during T cell development. So because SP1 and Notch1 are really antagonists, when Notch1 is there, SP1 cannot work very well. So we don't actually have that much information about SP1 in the single-cell seq data to make a network correctly based on that. Before going to our next steps or future work, I want to go back to our grand challenge that we have. Our grand challenge that we are trying to live with, scratching the surface of it, is to predicting the mechanism based on the form and function. And that's still a hard challenge, because when you see some phenotypic data or genotypic data, for example, single-cell seq data of your system, it is not truly telling you about mechanism. There can be multiple ways that the evolution can give rise to the same sort of function in terms of mechanisms. So for the next steps, we want to actually use IQCELL and then combine it in some sort of spatial transcriptomic, which has some sort of information about the signaling pathways in it, and then try to combine these two scales together to get a multi-scale model for developmental systems that you're interested in, that has both the gene regulation, also the signaling dynamics at the same time, and see how it works out. At the end, I want to thank my supervisor, Professor Peter Zanstra, and my colleagues and collaborations here, particularly Matthew Langley and Sarah Jane Dine, that was actually in Microsoft Research. I had the privilege to be there, and now she's moving to Google DeepMind. Thanks very much.
All right, great. Thank you, Tiam and Jason, for very interesting talks. We have some time for questions for both of you. So please go ahead. Anybody who has questions for either one of our speakers.
I'll go ahead and ask one.
Go ahead, please.
Yeah, thanks for those interesting talks. I have a question to Tiam. Is there a way to use your simulations to identify the most informative perturbations to determine gene regulatory networks? Or it could also be an efficiency question. What's the minimum number of perturbations you need to elucidate key features of the gene regulatory network?
Yeah, absolutely. In terms of a platform, basically, with one-click platform runs all the single and double and even three combination of genes perturbations, and it gives you the outcome. The question maybe that we can ask is, what are the informative perturbations that we can then go back to lab and perform, right? So basically, if we can predict some perturbations that are robust, and then go back to lab and ask them, definitely there's a way to doing it. Yeah.
John.
To get the naive optimist question out of the way, Can I interpret what you're saying to say that if I have the genetics of a virus and I focus on the spikes, that I can, in the computer, manipulate the genetic and explore possible mutations and look at the effect on the change in spikes, and then say, with this virus, here's kind of a critical path to the things that could mutate, so that we can anticipate some of the, to then look at it and saying, if that happened, look, this would be awful. But also, if that happened, we might be able to account for that in the design of the vaccine in the first place, so that the worst cases couldn't happen. Can you maybe actually predict the directions of the trajectory of the mutations of a component of the virus? And I'll accept, John, your questions don't even make any sense.
So, is that question for Jason, I assume, right?
That's for Tam.
Yeah, that was for you.
Oh yeah, sorry.
Where I mean, the other may have relevance, but I just locked on to a sentence that said, the effect of the gene perturbation on the developmental trajectory.
Yeah.
So does that mean you can look at manipulated, artificially created gene perturbations to determine ahead of time what change that could cause in the physical mutation? Is that what you were saying?
Yeah, absolutely. Sorry, the confusion was because the platform that we have is mainly specialized for developmental systems. So I'm not sure how we can really think about it in a virus. But if I want to again maybe reward what the title means, by genetic perturbations we mean basically at simplest level knocking down or basically overexpressing a gene, or in more advanced level, if you make a construct, like a synthetic construct, and you basically transduce yourself with it, which adds a new circuit to your system, basically predicts, the goal is to predict how it can affect the developmental trajectory of your differentiating cells, in our case, mammalian cells. So in principle, we are interested in, and we showed that we can up to some degree predict what's going to happen.
Okay, great, thank you. I have a couple of questions for Jason. You said in passing, it seems that you had an in vivo validation of your agent-based model. Did I misunderstand that? You didn't say anything further about it.
Oh no, I said we didn't do any direct in vivo validation. The closest in vivo validation we would have had was the cell culture data we collected that mapped very well to the model without doing any parameterization. Because we didn't do any parameterization of the model beyond the fibroblast data we used for the original model for training. But all the data you saw later on was not fibroblast data.
Okay, got it. And the other question I had is, I sort of missed that. What are we supposed to take away from this? What's the major takeaway from this stochastic interferon signaling and your model results?
Yeah, so that's very different from most of our studies, because most of our studies are much more applied, where we're looking very directly at either the response of males versus females or specific viruses versus each other. This is one of the few studies we did where it was much more of I would call it more of a fundamental study, trying to just understand simply, was there an advantage to having stochasticity or not? I would say that it does tell us that stochasticity does seem to be a major factor in at least determining what's going to be the population of dead cells in the end. So if you're looking to develop an effective model of a cell culture infection, you probably need to consider stochasticity as being one of the features of your model that needs to be included.
Okay, great. John Jin, you had another question.
Yeah, thank you. This is actually directed to Jason. A couple of questions just for clarifications. A very nice talk, by the way. You mentioned in your modeling, I think, primary interferon effects and secondary ones, where I think the primary ones were the ones that were infected by the virus and stimulating interferon, and secondary were the ones receiving the signals. So is there a role of autocrine, a role where the interferon produced by the initially infected cell actually has some effect on the virus production of that cell? Or are you looking solely at secondary cells that that were not infected being signaled by the initial cell?
So there is an autocrine and paracrine effect. So if you are an infected cell, you will have some interferon that will come back and bind the interferon receptor and upregulate interferon activity within that cell. We never tore the model apart to really do a comparison of how, if you took one cell that got infected, how did that rate of interferon production internally or activation of interferon stimulated genes more precisely compared to the actual rate of the viral infection within that particular cell. And that would have been something kind of interesting to actually consider. Maybe we'll do that following it up now. But for the most part, it has both capabilities, but we didn't really tear them apart any more than just looking at secondary versus primary.
Okay. Do you have a sense? I guess I don't know. I'm just curious, can an autocrine response be sufficient to shut down the production of virus in that primary cell?
I don't think so.
Okay.
I think typically once the cell has already gotten infected, it'd be too far ahead.
Too little, too late.
Yeah.
Yeah. Okay. Second question is, I think you were looking at interferon producing cells and percentages of those. I don't know if you're making some kind of distinction between potential defects in their ability to produce interferon, or, it could be also, I guess I'm thinking about the mechanism at which the signaling is either occurring, being generated, or being received. And, we can have defects in receptors, or we can have defects upstream. And I'm curious, are you making any distinction there? And is there a distinction to be made in which stochasticity could act differently in those kinds of scenarios?
So the specific mechanism leading to the stochastic behavior, we didn't try to dive into because within the couple experiments that we saw, there were a lot of variables we could have considered. So it could have been maturity of the receptors, essentially, maturity of the cells themselves, because of course the cell cultures, all the cells are in different cell states at any given moment. And that alone might have been a factor in determining which ones are actively producing interferon or not. And since that was just too many variables for us to look at, we kind of just grouped it all together.
Great, thank you.
Yeah.
Thanks. Any other questions? So I have a question for Tiam. So generally, algorithms like this to reconstruct networks from data, they're always underdetermined, they're vastly underdetermined. And so everybody who builds one of these, you have sort of a secret ingredient, a secret sauce that goes into it that makes it perform well on at least certain types of data. And in your description, I missed the part about the secret sauce. Maybe you didn't mention it, but it seems on the data that you showed, your algorithm performs very well. So what do you ascribe that to?
Yeah, I think that's a question that we are also asking from ourselves. So this work is on bioRCAV, also is under review, and we are now trying to test it on different data sets. Of course, the data set that we are working on are mainly in mammalian cells and differentiation of mammalian cells. So, but we are not trying to test it on multiple other data sets and test the performance. But I think there are few assumptions here that are really critical, and they might be correct or they might be totally wrong. One of them is the discretization of the genes into on and off state, and this is a really strong assumption because when you assign the gene to be either on and off, you're basically watching out many effects that can be due to the level of the genes and the basically kinetic. When you think about the gene regulation as like an ODE. Basically, based on the levels and the differential equations, there can be the effect of multiple. Like one gene can be at one level affect one gene and another level actually activate another gene. But when you assign on-off, you're basically washing out these type of things. So that's really important. And also it really depends on the quality of data set as well. So I think we were lucky because this data set that we used, it was really high quality. We know that single cell RNA-seq data sets can be really noisy, and particularly we are not interested in like all the genes that are important there. We are really interested in the transcription factors, and it's really well known that transcription factors are really noisy when it comes to single cell RNA-seq data, because the number of messenger RNAs that are expressed for them are really loud. So there's a high noise. That can be really, really important to quality.
Yeah, it's the discretization that you mentioned is really important. In our experience, we've had some data sets where we actually needed to, halfway through the time series, switch from one discretization method to another one. Otherwise, we would get completely crazy discretization results. The other question I had is, you seem to be looking actually at the developmental trajectories, and so that's where sort of timing comes in. Now, of course, Boolean networks don't have any inherent timing. So how do you assure that the timing inherent in your network makes sense?
Oh yeah, that's a great question. So basically, Boolean networks has a sense of the rough time, but they don't have the time scale, of course, and that's definitely the aspect that you're referring to. So we show that the cascade of events that we see in our Boolean network is recapitulating what the cascade of events when it goes to development, but we don't have information about the time scales. And recently in literature, you can find some nice work that they try to connect the Boolean networks to time scales by some sort of measurements that then you can go back and match to your network. But basically, the question that should we ask is how much information we can capture from time scales, and then if we import into your Boolean network, what can be the benefit of it? So in our case, we are just interested in the attractor, the endpoint of our development. But if you want to look at the trajectory and timing, definitely that should play into the network that we have. Yeah.
Thank you. Any other questions for our speakers? Well, if not, then I want to thank both the speakers, and I want to thank all of you for taking time out of your busy Thursday afternoon or evening or morning schedule, whatever it might be, where you are, and we'll see you hopefully again next week. Thank you. Bye.